I guess for the purpose of the presentation and the recording, even though we have a very small audience, I'll just go through, but stop me if you have any questions along the way. And I'll say that I've been at the NIH for 9 years, the National Institutes of Health, the Arthritis and Musculoskeletal Skin Disease Unit. I've been interested in immunodeficiency even before coming to NIH in my training, where I did some training in the lab of Jordan Orange, who's a very prominent immunologist, and that's really what got me started and interested in this. And I will say I have some late-breaking news that I'm actually going to be moving my lab to the Riley Hospital for Children in Indiana. So I have had an opportunity to get another position. I'll be the chief of the division and have a lab. So we will be continuing the work of seeing individuals with rare disease defects that affect the NF-kB pathway and others. And that'll be coming later this year. Okay, about this talk. So we're talking about NEMO syndrome and really more broadly on diseases that affect the NF-kB pathway. We'll talk about what these diseases are genetically, what they look like clinically. We'll talk about how they're diagnosed, and focusing somewhat on the NEMO syndrome, which is probably one of the better known of these disorders, but there's a lot of work that's being done now to understand other parts of the pathway and how mutations affect those pathways in humans. So we'll talk about how these other mutations also can lead to disease briefly, and then how the disease can be managed and treated, and what are the outcomes that we can expect. The other thing, just to state again, that this is a very informal session, so just stop me absolutely with any question that you have, because if there's something not clear, we can make it more clear. So we'll talk a little bit about what's called the pathway, the NF-kB pathway. And when we say pathway, what we're really talking about is this thing called signal transduction, sending signals from the outside of the cell, which I'm showing here. This is a sort of a sketch, a very schematic version of a cell. So the outside of the cell is the part in green that you see here, and the inside of the cell is the part in blue. And the nucleus of the cell is in white here, this round circle. And the one thing really to take away from this pathway, it's a signaling pathway, and generally it goes from receptors that are on the surface of the cell, or receptors that are actually inside the cell itself. And I'm showing some of them, but there's actually even others that I'm showing that I'm not showing. In the nucleus is where the DNA is, and if that gets damaged by UV light, for instance, or toxic chemicals, some that are used as chemotherapies, that actually activates this pathway too. So there's all kinds of ways that this pathway is triggered. And what I'm showing here is NEMO, and this is the paradigm molecule in the pathway, but also one that we'll talk about very briefly at the end is kappa-b alpha. We've done some work on one called A20. Learned about IKKI. Basically, all of the proteins that you see here, if mutated, and if the mutation is severe, can affect the pathway. So when we say that NEMO is a scaffold protein, what we mean is that it really binds to other proteins, it recruits other proteins, but its function is to be a structure that other proteins interact with. It's not an enzyme itself, so it doesn't make chemical changes to other proteins. By contrast, a protein, the kappa-b alpha protein, does have some scaffolding function because it does interact with this NF-kB itself. Its role is to is really important in activating this pathway. It gets phosphorylated. So what I'm showing you here in a little bit of a kind of like a pseudo animation here, but this scaffolding of NEMO, depending on which receptor gets triggered, NEMO will bring this whole group of proteins up to that receptor, and two other proteins. So NEMO, I'll tell you in a sec what the acronym stands for, but it's also called the gamma protein. But beta and alpha are very similar to it. Those are proteins that they're called kinases. They phosphorylate this I-kappa-b alpha protein, and that's important because this I-kappa-b alpha protein, once these receptors get triggered, it gets changed with this little UB tag, a ubiquitin tag, and then it gets degraded. So once it's degraded, that lets this NF-kB complex, it's actually a family of different proteins, but two of them will go down and be released to be able to go into the nucleus. And understanding this pathway is really important for us to be able to understand how different therapies might work or might not work in terms of what happens after this NF-kB bind. So it's all of the drugs that we choose to use or think that might work, or the therapies like a stem cell transplant, really are things that we can kind of rationally arrive at as options by knowing what happens when this NF-kB binds to DNA in the nucleus. So I'm showing just two little parts, two little segments of DNA here that it binds to, but actually this NF-kB binds to thousands of different places in the nucleus, different parts of DNA, so that different genes can be read. So this little arrow means that gene that's right there where that binds, that gene is getting read and made into a new protein. So some of these genes that get read are what we call pro-inflammatory cytokines, or cytokines are signaling molecules, kind of like hormones of the immune system. So these pro-inflammatory cytokines help us generate an inflammatory response or an immune response. So that's key. The other set of genes that are made when NF-kB binds to the nucleus are pro-survival genes, so that if NF-kB doesn't bind to the nucleus, you don't get these pro-survival genes made, and so the cell can die. And there's, I'm not showing here in this sketch, but triggering the TNF receptor, for instance, or sometimes triggering the toll-like receptor 3, those can trigger other cell death pathways simultaneous to this I-kappa-b alpha degradation pathway, so that if these pathways are triggered, but these death pathways are triggered, but the pro-survival pathways are not triggered, it can lead to cell death, and that can be damaging and lead to certain disease outcomes. So these two different pathways, pro-inflammatory and pro-survival genes. Another pathway that gets triggered by some of these receptors, and that NEMO is important for, is activating the antiviral response. So that this is showing another transcription factor, not NF-kB, this IRF3, and when that binds next to NF-kB, you get something called type 1 interferon made, and you get an antiviral response. So just to say, this is the pathway. It's kind of complicated, and by the way, I will send you this presentation if you want. Absolutely, yeah, no worries. So you don't have to write notes or anything, but
So on the antiviral
Yep.
response,
Mm-hmm.
so in Xavier's case, every time we stop antivirals,
Mm-hmm.
he has a relapse of chickenpox, varicella.
Really? Huh.
So every time we try to stop the antivirals, even though everything looks good transplant-wise,
Right.
every time we try to pull off of those antivirals, he reactivates.
Right. This would bear looking into. I mean, to understand how the I-kappa-b alpha mutation, that he has is affecting the antiviral response. We don't know, from other patients with I-kappa-B alpha mutation, much about varicella. In fact, I'm just based on just what I remember. We do have a patient with an I-kappa-B alpha mutation that we've seen who had a really big problem with Epstein-Barr virus.
Yes, I've heard about that.
And that caused what's called a lymphoproliferative syndrome, where basically the B cells, everyone gets infected with Epstein-Barr virus, and that EBV resides in our B cells long term. It just sort of stays dormant there. But in some people whose immune system can't sort of keep it dormant, they can get problems with just B cells expanding, and the whole response that triggers can be severe. And it's not cancer, it's not a leukemia or lymphoma, but it can look like it sometimes because you get just proliferation of abnormal B cells. Anyway, so that can be tricky. But antiviral response is important, and so that's interesting to hear about. And as we learn, we'll put the pieces together. But definitely, if just backing up on this slide, and because it's relevant to what to your son, if the I-kappa-B alpha can't get degraded normally in response to let's say a viral trigger, and so what I'm showing these squiggly lines here are in some cases viral nucleic acids. So we can sense that a virus is inside of our cell by receptors that sense not our DNA but viral DNA, and that triggers this pathway. Well, if the I-kappa-B alpha is not getting degraded, you would have a problem with making interferon, and have now this response is key. And so one thing to mention: some of these proteins that I'm showing here, for instance, A20 or TANK, TBK1 in some cases, these are actually what we call negative regulators. They're not parts of the pathway that trigger the response, but they're parts of the pathway that, even though they may help trigger, they also are key for turning off the response. And so, when we see a defect in one of these proteins, we might see a problem with activation of the pathway. We might see a problem with negative regulation. And so, we can see, it's not this simple as the next bullet shows, that immunodeficiency, you might think that immunodeficiency would generally be a problem with activation, and that inflammatory disease might be a problem with the negative regulation, because if you're not able to downregulate the pro-inflammatory cytokine production, you could see how that might happen. It's not that simple, but it helps to start thinking about some of these things. Just because there are sometimes not just one arm of negative regulation, but 3 or 4 that are each feeding back on each other in complicated ways. But I'll now just go on to talk a little bit about the different proteins in the NF-kB pathway that we know are related to disease in humans. This little Venn diagram here is showing that of the diseases in which there's, we think, too much type 1 interferon or an excess production, and of the ones in which there's impaired regulation of NF-kB, there is some overlap. So there's some that have both of that. There's some diseases that seem to be more related to just pure inflammation, triggering something called the inflammasome, although we are learning that some of these proteins also have to do with the immune response, but in very specific, select examples of a specific pathogen where you might need just the right amount of inflammation to really clear that pathogen. But then of the ones that affect the NF-kB pathway are the I-kappa-B alpha mutation, NEMO mutations, something called haploinsufficiency of A20, HA20, and then ones that affect 2 other proteins, otulin and a protein complex called LUBAC. And I'm not going to focus on those others. They all look slightly different in terms of the clinical phenotype, although many are associated with problems with antibody production, problems with NK cells functioning normally, and with problems with T cells. So we will focus on the NF-kB pathway. And so just to explain that the gene that codes for NEMO is called IKBKG, and this gene codes for the NEMO protein. So technically, it's the IKBKG gene and the NEMO protein. But if you say the NEMO gene, people will just know that you're talking about IKBKG. And this codes for this gene codes for this NEMO. It stands for essential modulator because it was found to be crucial for NF-kB activation. So if you don't have NEMO, you don't see NF-kB activation in cells. And then, just loosely we call the mutations in IKBKG NEMO syndrome, or something that also individuals with IKB alpha mutation can have, this hypohydrotic ectodermal dysplasia with immunodeficiency, HEDID or EDA-ID.
The cone-shaped teeth, the sparse hair, skin tissues,
exactly.
weird nails.
Yes, exactly. And so we can I'll just touch briefly on why that is that you see that. But basically, it's a combination of immune deficiency and developmental defects. So how do we diagnose this? We know the gene, it's the NEMO gene. How do we diagnose it? Well, it starts with the clinical history and the physical exam, because of course we wouldn't sequence someone if they had no problem with infections or they had no physical findings that were unusual. So it always starts with that. And we looked for what you just mentioned, conical teeth, or sometimes there can be missing altogether, or just maybe not conical, but something kind of not right with the teeth. A lot of tooth decay is common. Failure to either have them come in, but also sometimes retention of the primary teeth. And some individuals with NEMO mutation and with IKB alpha mutation have normal teeth. Absolutely would not trigger anything by a dentist. So that's unusual. Then we run lab tests, and these are basic lab tests, just cell counts. But as you know, sometimes the white blood cell counts are totally normal, and so you wouldn't know there was anything wrong unless you did special blood tests on the white blood cells, looking at the different percentages of the different cell types, like the T cells or B cells.
What are some of those tests that you'd be running?
Sure. So the main one that we run is it's called a flow cytometry test, which basically is a machine that has lasers, and you can basically tag every cell in the blood using antibodies. So we have antibodies to T cells and antibodies that specifically recognize B cells. By saying we, meaning humans have derived these, for research purposes, and so these different antibodies are triggered with a different sort of laser light, I guess, that really something that shines a different color when you activate it with laser. And so not only T, B, and K cells, but also we have antibodies to markers of memory. So when a cell becomes a memory cell, it will express a certain protein. And if it's a naive cell, it will express a different form of that protein. So we have antibodies to those two different forms, and so if we throw that antibody, or that, what we call it, a cocktail of antibodies, because usually we're adding 9 or 12, different antibodies, we run those through the cytometer, and so we can tell exactly the percentage of memory T cells, memory B cells. We can look on those memory B cells whether they also do or don't express another protein that might be important. So, that's how we learn.
That's different than chimerism.
Well, yeah. So that would be, a different type of test where you might sort cells and then look at, the DNA content of those cells. But actually, cell sorting is really kind of, sort of the big umbrella that cell counting is under. So you can sort cells, you can count cells,
Got it.
and by sorting them, you can see what what the proportions of chimerism there is. Yeah, and so. But we also do, as you know well, we check antibody levels. We check how these immune cells proliferate. When we stimulate them, there might be normal numbers, but if we, let's say, show these T cells Candida, and we would, expect there to be proliferation of cells because Candida is everywhere, and so we would have seen Candida. So, but if we don't see proliferation, we know that there's a failure of those T cells to recognize, the antigens normally. And so, anyway, this is all part of the immune function workup, and then that leads us to sequencing genes. And so, we used to sort of based on the history and physical exam and all the lab tests, we would say, okay, based on what we know, it's probably one of these 3 or 4 genes. Let's test these 3 or 4 genes. Now, what we tend to do is sequence all the genes and say, well, we still know what 3 or 4 we think it would be, so let's just focus in on those. But this is important to note because if a patient is found to have a gene mutation and we make a plan to treat them, but the patient doesn't do as we expect, then it makes us wonder why that happened. And what we can do is we can look back at all of the other genes that were sequenced at this time and see if there might be other mutations in other genes that are modifying the disease. Yeah, so
those results may be sitting out there. We just didn't
correct.
look because we went after.
Correct.
A small
We
group.
saw the obvious one, the
Right.
I kappa-b alpha, and we said, well, that's it. Let's treat that. And that would have been appropriate because it
Totally. He had
all the features and signs,
Yeah.
and he does have this disease. It's just that it's funny. Something else. And so we used to call these rare genetic syndromes monogenic. People used to say, this is a monogenic disease, to kind of distinguish it from what's called polygenic, where something like arthritis, where you have the bad luck of having 30 or 40 genes that each one, in a little way, creates the environment so that you all of a sudden get arthritis when you're 40. So we used to think, the monogenic ones, is due to that one gene mutation, but knowing that it really is modified by all the other genes. And sometimes it can be strongly modified by another gene. And so, it does always bear looking at if things don't go as we're expecting. Now, saying that, what do we expect? These diseases are so unbelievably rare anyway that we really are learning what the disease is as we're treating them. So, it's really not that accurate to say, we expect this kind of response. But still, with that said, it's worth looking at the other genes because even though, as you had mentioned, I mean, your doctors are astute. They know that if this is the clinical picture that we're seeing, well, there's other diseases where X, Y, and Z therapy work for that. We don't hold back on starting those other therapies without knowing that this person doesn't have a CF gene mutation. So anyway, so that's how that goes. And then this is always patient based. How we learn about things. It's humans who have disease who we're able to sequence, and then we can study this in different ways. So just talking a little bit more about the sequencing. These results are compiled from many different patients, many different sequencing reactions, and what I'm kind of showing you here is a gene on this axis here. It spans a certain length, and there's parts where when we sequence, we get a certain amount of signal, and the height of the peaks here just really kind of shows how much what's called coverage we have, how much sequence we got. So let's say, I don't see here how many individuals were sequenced to give this data, but let's say it's 50,000 different genomes were sequenced to give this data. You can see that on average, the very beginning part of this part of the gene didn't somehow get read as well as this other part right here. So we just have a little bit less sequence. It's still plenty to know what's called the reference sequence is. What is what's more common to have. But the one thing that's particularly tricky about NEMO is that we have a sort of a blind spot. So this is the same data for the NEMO gene, and you can see the very beginning of it we read fine, but we just can't see the end of it. Now the reason for that is because there's something called a pseudogene. So the NEMO gene has exons. That's the part that when it gets read makes a protein. And then the pseudogene has exons. They're in the reverse order, happen to be in the reverse order on the gene, but this doesn't lead to making a protein. So it got copied at some point back in our history as humans. It's just sitting there. But when we sequence, we have matching sequence from both the pseudogene and the gene, and we don't know. It's difficult for our computer algorithms to say, well, does this sequence go here? I don't know where the mouse went. Yeah, does this sequence go here, or should this identical sequence be mapped to there? So what happens is it throws it out. It says if you can't unambiguously say, just throw it in the trash, and then wait till we find a sequence that works. So there's ways around this that we have found. Sometimes what we have to do is we have to look at each individual sequence and find this is not totally related, but anyway, sometimes we have to look at each individual sequence and piece things together. It's very hard to do this in a standardized way, so we have to just go individually sort of patient by patient to look. But you can see here that if these are the individual sequences that I'm showing you in gray, in some cases you'll only find a mismatch in some of the reads, but not in others. This could be something that, for instance, in the case of chimerism, you might see, if it's not 100%, it would indicate that there's a mixed population of cells. Sometimes, without thinking about stem cell transplant, you can see, what we refer to as mosaicism, which is really similar, where an individual can have some of their cells have sequence with this sequence. Some of them have DNA with this sequence, and some of the other cells, a proportion of the other cells, have a mutation or a variant, and so that can lead to an altered phenotype. But the way we do that is by looking in more detail and using what is called next generation or high throughput sequencing. Other things that we can do is amplify the actual product of the gene. This pseudogene product won't amplify. So if we amplify it by PCR, and then we can purify it, first off we can see if there's two different forms. This form here skips out one of these exons, makes a different form of the protein, and we can find that it would be impossible to really detect this unless we use special techniques of analyzing sequence without the cDNA amplification. So now you mentioned the EDA. So it goes by a couple of terms. Either HED or EDA. Really mean generally the same thing. And there can be things other than hair follicles and sweat glands and tooth development. Sometimes we do see abnormalities of the nails. And then there's immunodeficiency. Variable. Usually. B cells are affected to one degree or other. I mean, it's really unusual to see fully normal B cells that make abundant antibodies of the right type. And so T cells, and sometimes the B cell number itself is down, not just the function. T cells and also can be abnormal in number and the memory phenotype. And the kind of bacteria that one sees is in susceptibility to is the encapsulated bacteria. So these are ones that often antibodies can really help protect against. But also we see atypical mycobacterial infection. So these, is that right?
The first
okay.
time you and I met, he was fighting a non-TB MAC infection.
Right. Okay. So right. So MAC is
We did an
really common.
18-month treatment of 5 different drugs.
Okay. 18 months, you said. Wow. And so was it cleared then following that? Great. And that's one thing. I mean, we touch a little bit later on treatment, but that is actually one of the most important things to be, if one is considering stem cell transplant, to be able to do that before MAC infections really take hold, and because it can be very, very difficult to clear, to eradicate. As an individual gets older, these don't get generally better; they get worse, and so that's important.
Yeah, it was 6 months of treatment before transplant, and he went through transplant, still on treatment, and then it took a year after.
Okay. And so, whether it's in the context of with stem cell transplant, or whether it's just in terms of optimizing a therapy because of other symptoms that are going on, definitely our approach is to really try to eradicate any microbe. So the immune response is always present to some degree or other, and as we'll mention just briefly later on, inflammatory responses can be exacerbated if some of these microbes aren't cleared. Anyway, the last thing just to mention here, we have seen opportunistic infections in individuals, both kappa-b alpha mutation and NEMO syndrome, and these would be ones that affect people that have really poorly functioning T cells usually. So pneumocystis pneumonia would be one that definitely is a possibility. And then there's prophylaxis for that. So, but the bottom point is that there's a wide spectrum. So even brothers that have the same mutation might have shown very different susceptibilities and patterns, and even other non-infectious phenotypes could be different. All right. So why did the ectodermal dysplasia happen? Well, you remember the sketch that I showed before of the immune receptors and how they signal. This is the EDA receptor. So this is not considered an immune receptor. It is important for the tooth and the hair follicle development. But this looks really very similar. NEMO is required in this pathway. kappa-b alpha is required in the pathway, and so that explains that, and that would be something that wouldn't really be affected by a transplant.
Right.
It's in the developmental stage, and what I will say though is that right. So let's leave that there. And so now, as I had mentioned, there's individuals without ectodermal dysplasia. We mapped it out, trying to see if we could understand if you could predict whether there would be ectodermal dysplasia based on the location of the mutation. In a really general way, you might say that if it's a mutation tends to be at this end of the protein, or maybe in this cluster, you might not tend to see as much ectodermal dysplasia. But to be honest with you, I think that it's really this same pattern we see with a lot of other signaling. So it's possible that just these regions here are sort of more tolerant of being changed than some of these other key domains. All right, and then the phenotypes are diverse. I had mentioned infectious susceptibility is really the main one that we are concerned with. Although I'll point out, and that infectious susceptibility is due to really immune cell receptor deficiency signaling that doesn't work normally. I don't have here, but an absence of lymph nodes is one of the things that we sometimes can see with NEMO mutation. And is that the case with Xavier? Okay.
So we were really surprised when we found the MAC infection because we found it in what few lymph nodes he has
Mm-hmm.
in his
Mm-hmm.
belly, but
Mm-hmm.
he doesn't seem to have very many
Right.
anywhere else.
Mm-hmm. Okay, so it could be that those were very small, but when they got colonized, or when they brought
Right.
them back there, they kind of grew, and there was some proliferation. But definitely, that's an important thing to look at and look for. And then I also will mention that autoimmune and autoinflammatory disease is something that exists with many primary immunodeficiencies, also with NEMO syndrome, and it's something that my lab happens to focus on. And so these are just showing, some of this phenotype of inflammatory disease. I'm just showing here all of the NEMO mutations that had been mapped. At one point when we made a large study to try to learn, what this disease really looks like, and you can see that there are some what we called hotspots at the time of different inflammatory disease or autoimmune phenotypes, and they did seem to cluster in two domains. One at this part, and really the mutations in individuals that led to this phenotype here at the end were ones where it basically led to chopping off of the end of the protein. So you didn't have that end there. It wasn't just a single letter change, so to speak, of the protein. These ones here are single letter changes for the most part, but we've been studying some others. I had mentioned previously that you can have different what's called transcript forms, where you splice out part of the gene, and there's a group of boys that we're following that tend to do that and have an inflammatory phenotype. So these are those mutations with autoinflammatory disease. Now, I'll mention it in another slide. We had found that mutations that affect this part of the protein, the C terminus, it's called, were ones that also didn't link up with a negative regulator of the pathway. So I had talked about negative regulators at the beginning, and that that's an important part of what NEMO does, the negative regulation too. And so we found that that those proteins don't bind this. Those proteins that have the end cut off don't bind this A20 protein, and that what that did, and I'm getting a little bit ahead of myself, but that caused increased signaling of this NF-kB pathway. Okay, but we also know that there are mutations that would be more similar, perhaps, it has to be studied in detail, but more similar perhaps to the kappa-b alpha mutation, for instance, that can't be degraded, where there's not increased NF-kB activation, but there's blocked NF-kB activation. And how can this cause problems? So this is something that's been looked at in mice, for instance, that have been made to lack NEMO in the intestinal epithelial cells. So I had mentioned before that you need NEMO to be able to activate NF-kB. So if you don't have the NEMO, what can happen? Well, this is a picture showing our the gut lining, and inside the lumen of the gut, within the tube, so to speak, of the gut, there's all kinds of gut flora, bacteria, that's normal to have, and it's also normal for us to be able to sense the presence of those with receptors that are on the surface of these epithelial cells. But what can happen if, and so the sensing of these happens through NEMO. So NEMO has to be there in the cell. If that's not sensed normally, what can happen is, again, these cells can trigger death pathways in response to activation, and so that can lead to bacteria getting further into the body than would be normal, and triggering an immune response because of some residual partial function of immune cells here within the body. And so that can lead to chronic inflammation by a defect. Now, I'm showing here the TNF receptor, and the way that we know that all of what's going on is partly due to TNF receptor signaling is that scientists have made mice that don't have the NEMO. They get intestinal inflammation. But if they make mice that don't have NEMO and don't have a TNF receptor, those mice don't get the intestinal inflammation, or they don't get the liver cell death, which the NEMO deficient ones get. So that's good evidence for that. And based on that and other work, individuals with NEMO colitis have been treated with TNF inhibitors, so drugs that are immunosuppressant that actually block TNF signaling, and they do much better. Now,
what are some of those drugs? Can you give me an example?
Sure. So one that's given by infusion would be infliximab, also known as Remicade. That would be the trade name. Humira might be one that you could consider, and then there's others. Adalimumab being Humira, the generic name. But and so definitely it's something that in someone with a primary immunodeficiency, gives pause, because we think immunosuppression bad. We must fight the infection, and we have some experience with using immunosuppression in individuals with primary immunodeficiencies, and it's tricky, but it works, and it works with caveats.
Right.
It's really important to know both the risks, but also how to have a heightened sense of monitoring for what comes up.
That with NEMO and an kappa-b issue, that there's kind of that risk imbalance in treatment of all of the cases, just because there's such a lack of number of cases to study, and I mean it's
right.
It's just kind of trial and error and watch and measure and look for all of those signs.
It kind of depends on sort of how you look at it and your philosophy, but absolutely every individual is their own case, their own disease,
Right.
Their own scenario. So it's definitely that's what we have to do. We have to try what we think will work when we feel that not trying something is too risky, because gut inflammation can lead to perforation of the gut, and that can be fatal.
Right, so benefits versus risk.
Yeah. And so untreated, it can be fatal. Treated, it can quote unquote fix things. And one has to keep monitoring and watching. Definitely, we have learned from experience also that we do our best to think of the best drug. Blocking, in a patient with kappa-b alpha mutation that we follow at NIH, who had bad problems with arthritis, we treated with an interleukin-1 blocking agent, interleukin-1 being one of these inflammatory cytokines, and the arthritis seemed to get much worse, and so we had to withdraw that medicine, and things were still looking pretty bad, and then we ended up finding that there was an infection of the joint. So what looked like only inflammatory and not infectious arthritis was infection. And I think that oftentimes you have a common, most, if not all the time, it's either an infection that's triggering the inflammation, or it's just an abnormal response to what you'd call colonization, or the flora that's triggering the response. But anyway, yeah. So we have to try what works, and just have to be very quick to look and see what's going on if things aren't working. Okay. So I had mentioned something about these C-terminal truncation mutants. just maybe to not spend too much on this part here, but we looked at all the individuals with a C-terminal mutation compared to ones that had what's called a missense mutation, so just a single letter change in that protein. We found that really it was the absence of that part of the domain, because that has a certain property. Well, we looked at whether it could bind to other negative regulators. This is the part that I had alluded to before. But then that opens us up to sort of understanding some other diseases. So at the same time that we were working on this, we were aware some folks at the Genome Institute and NIH were looking at individuals with this mutation in this A20 protein, and so we were able to study them. These are some of those individuals, and we found that based on that, this interaction is very important. So that's just work that we're going to be looking at in the future. How these proteins interact, and so in an individual, just to broaden this a little bit, with an I-kappa-b alpha mutation, for instance, in whom we'd expect impaired NF-kB activation. Now, I say expect that. We don't know until we look, experimentally, because we can predict it. But we have several examples where a mutation leads to a prediction that the pathway would be defective, but rather instead we find that the pathway is overactivated. And then we have to do some more studies, and we were able to figure it out, but it doesn't always follow what you predict. So anyway, in a case where there's an I-kappa-b alpha mutation, generally you might expect impaired degradation, impaired activation of NF-kB. Well, A20 happens to be one of the proteins that's most highly upregulated in response to NF-kB activation. A20 is highly upregulated, as is I-kappa-b alpha itself. But anyway, so in an individual with colitis with I-kappa-b alpha mutation, we would certainly want to look at A20 levels, and if we can learn by studying some of these molecules how to kind of potentiate the A20 NEMO interaction, that might be a potential therapy for an I-kappa-b alpha colitis, for instance. That's why I just mentioned my work at it. So just briefly, other individuals with autoinflammatory disease, in particular, eye and some central nervous system bleeds, also with some skin inflammatory skin changes, something called paniculitis. They have mutations not in the coding part of the gene, that's always, one of the patients did, but they had mutations in a part that is really important for splicing of the coding parts. So this sequence here that I'm showing you doesn't actually end up coding for a part of the protein, but it leads to sort of stitching the different parts together. Anyway, so that's why we look in more detail, and that's when we do some of this sequencing. So these individuals happen to have increased susceptibility, at least their skin fibroblasts, to viral infection. I'm showing here a virus that makes a green protein when it infects cells, and so on the bottom are cells from the patient that got infected. This is actually parainfluenza virus that causes severe respiratory inflammation in neonates and in young children, and also it can infect immune cells, T cells. But what we found was that in this case, TNF stimulation of these cells really helped these cells to not be infectable with this virus, and even much more so than the healthy control cells. So there's something weird with this particular mutation from these three boys that splice out this form of the NEMO. And so, looking at that, so these are skin cells, skin fibroblasts, that we think might function similar to epithelial cells, generally, maybe different than immune cells. So here's T cells from a patient with this mutation compared to another patient with another NEMO mutation that's a loss of function mutation. This one doesn't get infected with a virus as well. So there's this big difference, and that's an important point with the I-kappa-b alpha also in the NEMO and many of the other mutations in this pathway, that epithelial cells or skin cells might have different signaling characteristics based on a particular mutation compared to T and B and immune cells. So that's one of the things we study there, and it really impacts. This difference does impact how we think about, for instance, stem cell transplant as a therapy. So I won't go into the details of this mechanism, but maybe just to stay on here. So when we think about therapies, there's the potentially curative therapy of a transplant, a stem cell transplant, in general in immunodeficiency. Now, there's some immunodeficiencies where we, in quotes, know that immunodeficiency is due to a defect in an immune cell and an immune cell only, because the gene that's mutated is only expressed in immune cells. It doesn't make a protein in any other cell. So if you can give a person a new immune system that has the right gene sequence, they are, in quotes, cured. I mean, there's always
for gene therapy, and it comes into
play. Right. Now, so the thing is different in the case of these NF-kB pathways, because we know the protein is involved in other cells that are not in the immune system, or that are in cells that have some immune function, but they're not TB, NK cells, macrophages, monocytes, that kind of thing. So they might be cells that live in the thymus, where T cells, mature. They might be cells in the lymph nodes, as we mentioned, that won't get fixed by a transplant. So, and again, like you had mentioned before, these cases are unique. We don't have enough numbers yet to know. Although I will say, in general, there have been some larger studies recently looking at many patients who have undergone stem cell transplant. There are some general concepts we've been able to take away. One is that if we see severe mycobacterial infection really take hold and be present for years, it makes it much more challenging to have a successful transplant. The other is that we really use some of the lab studies occasionally to try to gauge how severe the mutation might be. Some individuals with NEMO mutation really don't have enough infections or enough of an inflammatory phenotype to make a transplant, stem cell transplant, reasonable. It's not warranted. We can that's one treatment, but others, that I mentioned, cytokine blockade, immune suppression, also just as important are prophylactic antibiotics and replacement therapies. For instance, the IVIG or subcutaneous immunoglobulin. So expectations. The more we know, the more we learn, the more we'll be able to do. Definitely, I think that the last 5 years of stem cell transplants for NEMO syndrome, I-kappa-b alpha, and similar disorders has shown that it's very unlike the 20 years before that, or the 80s, for instance, or the mid 90s. That outcomes are better, and so there's less hesitation if we have evidence that the mutation might be severe to do that. Now, we're obviously not thinking about that in all cases. In some cases, there might have been a transplant, and there are still symptoms or new symptoms, and so these are worth continuing to investigate. And if we can choose the right anti-cytokine therapies to target certain pathways, those can be key. It's very tricky if there's a lot of infection burden. All right. So just to mention that there are other phenotypes than what we've mentioned that we are interested in understanding, and that for now, kind of are in the process of being worked out only. There are some central nervous system changes, some cognitive changes we've seen in some individuals with I-kappa-b alpha mutation, such as epilepsy and seizures and epilepsy, and others having white matter changes that look like multiple sclerosis. Others who've seemed to be in that category, but who we have found to have an infection with an opportunistic virus, in the brain. So things can look very different. But anyway, we're making models in mice to be able to study these. And so phenotypes are some things we're always collecting. So these are, just to end with, individuals with I-kappa-b alpha mutation that, showing here in red, that have an inflammatory disease phenotype. And we, really are just at the early stages of trying to understand exactly how and why this happens. Now, there can be obviously the combination of infectious susceptibility and inflammation, and so this is something that really working between human samples and animal models will be able to understand. Okay, so that concludes this part. We can, I can take any other, additional questions, or we could also just, you have my email. And we can set up a time to talk if you have other, concerns or issues. I can also get in touch with doctors at, where your son's been treated. That's always something I'm very happy to do.
That would be helpful. I think, one of the questions I have is, so we have a new symptom that we've never dealt with.
Mm-hmm.
And he's got what look like holes in his nails.
Okay.
That are starting to show up.
Pits, are they sort of like indentations, impressions, or actual hole all the way through?
Actually, almost a hole all the way through.
Mm-hmm. That might be. I've not seen, severe nail pitting, but
yeah.
This is something we see. Partial forms with certain kinds of joint issues. Certain kinds of arthritis tend to also be associated with nail pitting. We can think about that. And now, are there any other issues with the nails? Is that leading to other problems?
It's not.
Or it's
just all of a sudden started to show up. So what looked like just a little pit in the nail, has grown to be actual hole.
Right.
Like the size of the tip of a pencil.
Wow. Okay.
And then we're having weird days where he'll say that his legs hurt and he doesn't want to walk. I'm assuming we went through a period of that when he was first diagnosed before transplant.
Right.
And we thought it was beginning signs of arthritis, which I know are really common in NEMO and the I-kappa-b. And then it kind of resolved itself and he didn't have issues for a long time, and now we seem to have days where it kind of creeps up.
Okay. Well, yeah, I would say, generally, only way to know is to examine the joints, to have one of the immunologists, but usually rheumatologists are more used to looking at every single joint and knowing the full range of what's normal, and that would most likely be worth it.
Maybe worth it to go back.
Yeah, there's a lot of non-arthritis joint pain that one could have if there are ongoing infections.
Well, his inflammation markers have been high for the last few months.
Right.
So,
and with infection, they will be.
Right.
So what can be done is the joints specifically can be looked at. They can be examined. They can be imaged. Fluid can be sampled from within the joint. Numbing medicine is put over the joint, and a needle is put in, and just the fluid that's taken out is looked at. Our individual with I-kappa-b alpha mutation had arthritis for a while, And it turned out that when we put a needle in and cultured the fluid, we found that it was a mycobacterial infection. So it was an infectious arthritis. And these infections don't look the same in an individual with an I-kappa-b alpha mutation to other individuals that don't have the mutation. Meaning,
Okay.
it just...
Right, like his MAC infection wouldn't have been found. We just happened to see one lymph node that was a little bit bigger than what it
Right.
had been in previous scans,
Right.
leading up to transplant.
And it was sampled.
And at the very last
Mhm.
minute, we literally were like, it was the day before
Right. Wow.
chemo was supposed to start.
Really? Mm-hmm.
And yeah.
And what happens?
Dorothy just had this moment of like, you know what, I
Let's-
think I want to look at that lymph
right.
node before we go any further.
Right, right,
And so that's what showed us the MAC infection.
right.
We ended up on clofazimine,
Mhm.
I think is the drug that we ended up getting that hadn't been released yet.
Mm-hmm.
So, going through that whole like FDA
Mm-hmm. Mm-hmm.
compassion relief
Right, right.
to do it, but it seemed to have worked. Now
Mhm,
it hasn't
mhm.
shown up, but it's interesting that you say that there's still, that there's always a possibility that it's not going to present like it would.
Right. Well,
So
we
I think it's good to just be aware.
Yeah.
Like
it's funny. We talk about this with the infectious disease group at NIH a lot, and I think I have a little bit of a reputation amongst them of being the one who always thinks that there could be a bug in there. I mean, which is weird because they are the infectious disease doctors, and and they are like the world best at that,
Right.
And I'm the guy who's always screaming, I think there could be a bacterium in there. It's kind of it happens in medicine sometimes that the rheumatologist tells the kidney doctor, I think there's a kidney problem with this person. That's the main thing, and then the nephrologist says, no, it's an autoimmune thing. That's the main problem. Anyway,
like
but
kind of how it goes at UCSF.
Yeah, so, and they're right most of the time. Sometimes though, weird stuff is found. It's tough.
Are there active studies going on
Mhm.
at NIH within NF-kB, the I-kappa?
Yeah, so there's apart from my group, there's another group that's looking more at these for the allergic diseases, and how this one protein A20 a little more than the others might lead to allergic responses. I think that definitely at the Genome Institute there's Dan Kastner has a lab and they have worked on this otulipenia that I mentioned very briefly. Mm-hmm. But not so much the NEMO and not I-kappa-b alpha, although certainly those patients that I mentioned in that slide at the very end, I'm trying to think about the second one, how we learned about her, but one, if not two of them, were seen by the Kastner group. And then they got me involved. So I think that they'll continue to, I don't know if they're going to going forward, look into that. But as it's been working out now, it's been mainly my group and NIH. But we're gonna continue that at Indiana University School of Medicine, and I assume there might be. It's funny. There's the infectious disease group, Steve Holland is the head of the lab that oversees that and the clinical service there. They have patients with they see the patients with I-kappa-b alpha mutation that come always. But as far as working on the mechanisms, I don't think there's any other groups. There's a few groups in scattered in France. There's a group.
I think that's where the other patient, the older one with an I-kappa-b. gain of function mutation. Like his, if I remember correctly, his mutation and my son's mutation are almost identical.
Okay. Mm-hmm.
And so I know that we've mimicked a lot of like treatment-wise based off that case.
Mm-hmm. Mm-hmm. Yeah, like so what's worked for them has been tried here. Right. Yeah, there's definitely there's a few of them around. There's a Finnish patient, a Spanish patient, an English patient, and those have all responded well to immune suppression.
Okay.
They've had different things though. Colitis has been a big one.
Yeah, it's just starting to kind of rear its head.
Okay, yeah. And then sort of brain neuroinflammation is another one. And then there's this thing called sarcoid, which basically affects the lungs and joints and can affect other things too. Could affect the gut. But you get these things called granulomas. Granulomas are like big cluster balls of immune cells and these sort of epithelial type cells. But anyway, that responded to immunosuppression. But I guess the thing is just to try to have everyone communicating on clinical outcomes and what's worked on these rare cases.
Yeah, I think one of our challenges right now is that our teams are all kind of on different pages.
Right.
So I have GI and pulmonology who are like, maybe we should look at trying immune suppression stuff. And then I have immunology who's like, no, we don't think we should.
Right.
I think it's just because, he has a complicated case.
Right.
He's the only one that they have, right? I think that's where it gets tricky. It's not like...
No,
I'm sure
there's no obvious
it's
answer
not like
either.
You have 40 patients with this condition.
No.
You're dealing with a one-off out of however many patients you're treating, and so I think there's a little bit of a level of I don't want to say fear, but just the unknown.
So much uncertainty.
Yeah.
And there is this rule of, first off, don't do harm. Don't do something that's going to make things worse.
Right.
And so, but that's balanced by, well, there's terrible stuff going on, and what are we going to do about that? This is one of the trickiest parts, I think, immune deficiency and autoimmunity or autoinflammation. One thing that's guided us at times is trying medicines that we know have what's called a really quick off rate. That dose the medicines are dosed daily. So if you start seeing, and it's out of the system. But it might not be the right pathway. It's like, okay, great, it's a short off, but it's not the right pathway.
Right.
So it's very challenging. I mean, we've just seen that with some of these, it's looking back, it's easy, but looking back, we've seen that for some, they've really responded to TNF blockade is the main one we've tried.
What are your thoughts on gene editing in the future as NEMO and kappa-b?
Well, it can be a cure, down the road. What it would require, though, it's tricky because even gene editing, if it's a particular, very specific, limited tissue, let's say there's a gene that affects liver cell function. Well, you can target only liver cells to get edited. And you might also have a gene that affects retina function in the eye, so you can target just the retina. Great, through gene editing that way. Most of the gene editing, as you know now for immunodeficiencies, has been peripheral blood immune cells that then get put into a patient. So it will.
I think the big question with NEMO and ICAP is the cells through the GI tract and the cells that can't be touched.
Exactly. Just like with hematopoietic stem cell transplant. Because if you want to think in a little bit of an unusual way, in a way that's gene editing,
Right.
It's kind of, well, it's changing the gene
Right.
to one that's fixed, even though you're not. It's editing by cut and pasting in a way, not by hitting that back button and actually editing it. Yeah, it'll happen someday, but we're just at the very beginning parts. So I think that the best I can say now is that for someone who's having symptoms that really seem to be not due to primarily immune cell function, but that could be due to resident cell function. Some of these cytokine blockade therapies would be worth thinking about.
Okay.
Yeah.
I'll have the team reach out again.
Sure, and I'll look through my emails
Okay.
and see. Yeah, absolutely. We can only see if we can help with an
Right.
idea or say what we know, what we've seen.
Well, my original thought was just to, maybe even just be able to get you the blood work that you wanted originally,
Oh.
since we couldn't do it, right?
Right.
I know you had given them a list of samples that you wanted,
Right.
and we were too close to transplant to
Yes.
pull that much.
Yes,
So,
yes.
even if that's just a starting point to look at something that they may not have looked at.
Right. For sure. And the other thing we could do, if it can be done, if he's well enough to provide a skin biopsy sample,
Oh,
because we can grow those cells up, and we study those in culture. The other thing we can do is take a swab of nasal, and get the epithelial cells and
Okay.
culture them. So, if there are respiratory issues and we can see that there's defect in epithelial cells, respiratory epithelial cells, I mean, they're not identical, the ones that
Right.
belong to there, but it
Right.
gives us some clue.
Right.
Yeah. So we can still learn things by, by studying, his cells.
Okay.
Yeah.
Thank you so much.
Sure, my pleasure. Thank you for staying awake during the talk.